Okay, guys, today we are going to look at glycolysis. Glycolysis in biochemistry, or the breakdown of glucose in the body of the living organisms, humans, animals. So, after your hard day's work, you come back from work, you eat. The food that you eat has to be breaking down. So, we are looking at the pathways in which the food that we eat will be breaking down and it will be utilized in the body. So now today we are looking at the utilization of the glucose in the body. How does the glucose that we eat be utilized in the body, and how is it useful to the body? How does it be utilized in the body? That's why we are going to look at a pathway called glycolysis. So please, this is OBED'S tutorials. Kindly subscribe, share and follow for more updates on anything in biochemistry. So glycolysis is just a series of enzyme catalyzed reactions, that convert glucose to pyruvate by yielding specific amount of energy in the body. That is glycolysis. We eat the glucose will be breaking down, then it will produce energy. That's the process. So we are going to look at how and when does the glucose produce energy. What are the processes involved? Okay, so after eating our meal or any food containing glucose, the glucose has to be transported from the blood, then it gets to the cytoplasm of the cell. That's where the glycolysis takes place. So now let's look at the definition of glycolysis. Glycolysis is a series of enzyme catalyzed reaction that occurs in the cytoplasm or the cytosol of a cell, which involves a stepwise breakdown of glucose to form pyruvate with a net gain of 2 molecules of adenosine triphosphate, which is ATP, the energy, and the formation of NADH, which is nicotinamide adenine dinucleotide molecules. So we break down the glucose to form the pyruvate, but it involves steps, enzymatic steps or reactions to yield the pyruvate. So we are going to look at the steps that involves in glycolysis. So now, okay, we have it, the steps in glycolysis. The first step is phosphorylation of glucose to produce glucose phosphate by an enzyme called hexokinase. So after the glucose is transported to the cytoplasm of a cell or the cytosol of a cell, remember glycolysis takes place in the cytosol of a cell. The hexokinase, what it does is it will transfer the phosphate group from an ATP to the glucose, so specifically to the carbon 6 of the glucose molecules, to form something that we call glucose phosphate. Okay, that's what takes place in the first step. And going forward, the glucose phosphate form has to undergo another enzymatic reaction to form something that we call fructose phosphate. But mind you, from glucose to glucose phosphate is a committed step in glycolysis. Why is it a committed step? It is a committed step because after the glucose is transported to the cytoplasm of the cell, it is converted to glucose phosphate. This glucose phosphate cannot penetrate the cell membrane and go back or go out from the cell. So meaning that the moment the glucose phosphate is formed, it creates an impression that by all means glycolysis will take place. That is what we mean by a committed step. So we are on the second step. So glucose phosphate has to undergo changes, isomerization, to form a fructose 6-phosphate. So mind you, glucose phosphate is an aldol. The glucose is an aldol sugar. Then the aldol sugar will undergo certain isomerization to form fructose, which is what? A ketone sugar. You get it? By an enzyme called phosphoglucose isomerase. Phosphoglucose isomerase. So what happened is that the carbonyl oxygen of glucose phosphate is shifted. Carbonyl oxygen of glucose phosphate is shifted from carbon 1 to carbon 2 by the enzyme called phosphoglucose isomerase to form what we call fructose phosphate. That's the second step. And we come to the third step in glycolysis. So the third step will just convert the fructose phosphate to fructose 1,6-bisphosphate by an enzyme called phosphofructokinase 1. So now we talk about the committed steps in glycolysis. The first step is a committed step, and then the third step is also a committed step. By the third step is the main committed step, meaning that is the main thing that would determine the occurrence of glycolysis in the body. So now the enzyme that we call phosphofructokinase 1 will add a phosphate group from ATP, another molecules of ATP, to the fructose phosphate, creating two molecules of phosphate, okay, on carbon position 1 and carbon position 6 of fructose molecules to form fructose 1,6-bisphosphate. Okay, yes, it is a main committed step in what? Glycolysis. So you see, in the first step we use one molecule of ATP, and then the third step we use another molecule of one molecule of ATP, meaning that in glycolysis we invest 2 molecules of ATP. So step 1 to step 3 is what we call the investment stage. Because we invest certain amount of ATP. You get it? So, and then step 4 we call it step 4, 5. We call it splitting stage. That is where we cleave the fructose 1,6-bisphosphate that is formed from step 3 to form two molecules of different compounds. We have dihydroxyacetone phosphate and then glyceraldehyde 3-phosphate by enzyme that we call hydrolase. So this hydrolase will just cleave the fructose 1,6-bisphosphate into dihydroxyacetone and then glyceraldehyde 3-phosphate Okay, so this is step that we call a splitting stage. We split it to form two molecules to form my dihydroxyacetone phosphate and then glyceraldehyde 3-phosphate. So the glyceraldehyde 3-phosphate is the only structure or molecule that can proceed in the glycolysis process, and dihydroxyacetone cannot proceed in glycolysis as well. So it needs to be converted to glyceraldehyde 3-phosphate for it to proceed in glycolysis process. So we have two molecules of glyceraldehyde 3-phosphate from the breaking down of the glucose. And step 6. So step 5 is when we isomerize the dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. So we form by an enzyme called triose phosphate isomerase to form two molecules of glyceraldehyde 3-phosphate. So let's go to step 6. Step 6, we have the oxidation of glyceraldehyde 3-phosphate to form 1,3-bisphosphate glycerate by an enzyme called glyceraldehyde 3-phosphate dehydrogenase. So now for this step, we oxidize. This is when we introduce the reducing equivalent, which is NADH. We oxidize the glyceraldehyde 3-phosphate to form 1,3-bisphosphate glycerate. 1,3-bisphosphate glycerate. This step is oxidation though, but it also involves the transfer of phosphate groups to the glyceraldehyde 3-phosphate to form 1,3-bisphosphate glycerate, converting the NAD plus to NADH. This is why we produce one molecule of reducing equivalent by an enzyme called glyceraldehyde 3-phosphate dehydrogenase. Alright, so let's come to step 7. Step 7 is the formation of 3-phosphoglycerate by yielding energy, specifically ATP. And this is catalyzed by enzyme called phosphoglycerate kinases. So now 1,3-bisphosphate glycerate that will form from the step 6 will be converted to 3-phosphoglycerate. This process is what we call substrate level phosphorylation. We produce ATP from the substrate that we have. Okay, produce the ATP from the substrate that we have. How do we produce the ATP? One phosphate group from the 1,3-bisphosphoglycerate is added to ADP in the process to form one molecule of ATP, which is this process we are just using one molecule of glyceraldehyde 3-phosphate. Mind you, we produce two, so we are using one for this. So at the end, we have to get a maximum number of 4 ATPs produced at the end of the glycolysis. So 3-phosphoglycerates will be produced. We yield our energy, process we call substrate level phosphorylation. Also, step 8, we have the isomerization of the 3-phosphoglycerate that is produced in step 7 to 2-phosphoglycerates by an enzyme called phosphoglycerate mutase. So for this reaction, it's just the interchanging of the position of the phosphate group So we interchange it. We change the position of carbon 3 to the position of carbon 2 to form 2-phosphoglycerate. That's it. We just change the or transfer of what the phosphate group from carbon 3 to carbon 2 to form 2-phosphoglycerate by an enzyme called phosphoglycerate mutase. And also we have step 9. Step 9 is where we dehydrate 2-phosphoglycerate to produce phosphoenol pyruvates. We dehydrate 2-phosphoglycerate to produce phosphoenol pyruvate. So we just remove a water molecule from 2-phosphoglycerate by enzyme called enolase to produce what? Phosphoenol pyruvate. So when this phosphoenol pyruvate leads to formation of the pyruvate by enzyme that we call pyruvate kinase. So what pyruvate kinase does is it will remove the phosphate group from phosphoenol pyruvate to form 3-carbon compound that we call a pyruvate. So this phosphate group removed from phosphoenol pyruvate will be added to ADP to form another molecule of ATP. So now step 7, we form molecule of ATP, one molecule of ATP, and step 10, we also form another molecule of ATP by process that we call saturate level phosphorylation. So at the end, one glyceride 3-phosphate produce 2 ATP. Mind you, after splitting a glucose, we produce 2 molecules of glyceride 3-phosphate. If one produce 2 ATP, then 2 molecules of glyceride 3-phosphate will produce 4 ATPs. And starting of this process, we invest 2 molecules of ATP at step 1 and step 2. So after we subtract the investment, the energy that you invested in the process, you will get net 2 ATP from glycolysis. Okay, so this is the overview of everything. This is the overview of whatever we've done. The glucose will be converted into glucose 6-phosphate. So the energy invested, ATP is invested. A phosphate group from ATP will be added to the glucose. So ATP will change from triphosphate to diphosphate. You get it? By enzyme called isokinase. Also, the glucose 6-phosphate will also undergo isomerization. Glucose is aldose sugar and fructose is what? Ketose sugar. So the glucose will isomerize to fructose by enzyme that we call phosphoglucose isomerase. And also step 3, this fructose 6-phosphate we also invert another molecule of ATP. So the ATP will add one phosphate group to the fructose 6-phosphate to produce fructose 1,6-bisphosphate. That is energy investment stage in glycolysis. When we come to step 4, it's a splitting stage. We split the fructose 1,6-bisphosphate into 2. We've split it into glyceride 3-phosphate and then dehydroxyacetone phosphate. So glyceride 3-phosphate will undergo isomerization, as we said earlier, to form 1,3-bisphosphate glycerate. So mind you here, it's oxidation though, but we introduce a new molecule of what? Phosphate group. Okay, that's why we get 1,3-bisphosphate glycerate. So we invest 2 molecules of what? NAD plus to yield. So NAD plus is oxidation state, so it will be reduced to what? NADH. So NADH is reduced NAD plus is reduced to NADH, forming hydrogen ions. Okay, and also step 7. This 1,3-base phosphoglycerate will undergo a step that we transfer a phosphate group from the 1,3-base phosphoglycerate to ADP here to yield ATP, and also lead to the formation of 3-phosphoglycerate by an enzyme called phosphoglycokinase. And come to step 8. Step 8 is phosphoglycerol isomerase, phosphoglycerol mutase. We just convert or transfer the phosphate group from carbon 2 to from carbon 3 to the carbon 2. That's we interchange the position on phosphate group. And also step 9, we just convert the 2-phosphoglycerate to phosphoenol pyruvate, that's PEP. So it's a dehydration process. We dehydrate it by producing molecules of water, okay, by an enzyme called enolase. And the last step is also a substrate level phosphorylation, where the phosphoenol pyruvate is converted to pyruvate by an enzyme that we call pyruvokinase. This one to yield an ATP. So the phosphate group on the phosphoenol pyruvate will be added to adenosine diphosphate to form adenosine triphosphate. Thank you very much. Please, this is OBED'S tutorial. Kindly follow, subscribe and share. Okay, see you next time. Bye bye. So watch out for my next video on calculations of ATP in glycolysis, and also the process that involve in electrons transport chain. Thank you very much. See you.